Hello friends, my name is Arfat Hussain and I am from Kashmir. I have created YouTube channel just to help the students, just to help the medical and paramedical students in clearing their doubts about the different topics, about the different disorders, we can say. And I hope this channel will help the students and clear their doubts as soon as I can. So this is my first video, so I had no suggestion from anyone that which topic I should deliver first and which topic I should do the next. And I decided it by myself that which topic I should do the first, and you can give me the suggestion for the next topic. So the topic that I decided today is about the DNA, is about the DNA formation and about the DNA structure. So let me give a little intro about the DNA. DNA contains all the life programs. All the life programs are primarily stored in the DNA. All the life programs are primarily stored in a molecule of DNA. At the time of fertilization, when the ovum is fertilized, at that time, the fertilized ovum, the genetic material that it contains the information, and that information leads to the formation of structurally and functionally organism. And we can say the plan of life is present in a molecule of DNA, or we can say the master plan of our life is the DNA molecule. DNA expresses itself in the form of RNA. The functional copies of DNA is RNA. You will come to know about it later. I will explain one by one, don't worry about it. So the functional copies of DNA is RNA, because RNA leads to the formation of proteins. Proteins are of two types: structural proteins and functional proteins. Structural proteins gives the structure to the cells, and functional proteins regulates the function of the cells. So DNA gives rise to the RNA give rise to the proteins, and proteins are of two types: structural and functional. So the structural proteins maintains the structure of the cells, and the functional proteins regulates the function of the cells. Correct? So the type of proteins present in the cell determines the type of cell. Correct? And the type of cell determines that what type of organism it's going to be. Correct? So the combination of cells forms the organ. As you know, this is the basic, and assembly of different organs makes the organ system, and the assembly of different organ systems makes the organism, complete organism. This is just a little intro about the DNA. Now we are going to discuss what is DNA. Introduction is over. Now coming to the DNA. What is DNA? If we ask to anyone what is DNA, they will say deoxyribose nucleic acid. But that is not the definition of DNA. That is just the full form of DNA, or we can say the chemical name of DNA, we can say, right? DNA is a linear polymer of nucleotides. That is the exact definition of DNA. DNA is a linear polymer of nucleotides. Now the terms are coming here: linear, polymer, nucleotides. So linear means unbranched. Any chain that's having no branch is linear. So we can say DNA is an unbranched polymer of nucleotides. So linear I have covered now. Linear means unbranched. The chain that's having no branch is linear. Correct? Now coming to the polymer. Polymer means combination of many units. Poly means many, mer means units, as you know. So combination of many units is known as polymer, right? So this is the unbranched combination of many units of nucleotides. Now what are nucleotides? That I have to discuss in detail, because there are many things in the nucleotides. There are many units in the nucleotides. So polymer you got, linear you got. Now there is the turn of nucleotides. So if we say what is the raw material of DNA, or we can say what is the raw material of formation of DNA, or building blocks of DNA. The building blocks of DNA, or the raw material for the formation of DNA, is nitrogenous bases. They are the nitrogenous bases that are required for the formation of DNA. So nitrogenous bases means what? Now this is the another term. After clearing all those terms, you will come to know about nucleotides. What is nucleotide? You will come to know after that. So first you should know what is nitrogenous base. Now nitrogenous bases are the nitrogen compounds containing carbon rings. Simple. Nitrogen compounds containing carbon rings. Simple. Or we can say nitrogen containing carbon rings. They are the nitrogenous bases. So nitrogenous bases are of two types: double ringed and single ringed. Double ringed are known as purines and single ringed are known as pyrimidines. So nitrogenous bases are the nitrogen containing carbon rings. Nitrogenous bases are of two types. One is single ringed, another one is double ringed. Single ringed is known as pyrimidines and double ringed is known as purines. Correct. Purines contains two types of nitrogenous bases, adenine and guanine, and pyrimidines contains two types of nitrogenous bases, and they are cytosine and thymine. It's not actually thymine, it is thymine is a vitamin, and because it's thymine, so it is a thymine, not thymine. Adenine, guanine makes the purines, cytosine and thymine makes the pyrimidine. If we see the structure of DNA, then still there is lag. Still it's not enough for making the DNA. We need to make the nucleotides first. We don't have the raw material enough for making the DNA still. So we need to fuse these nitrogenous bases with the sugars. What type of sugars are present in the DNA? They are the pentoses. Simple. Sugars containing 5 carbons. So for making the nucleotide, we need to fuse the nitrogenous bases with the sugars, and those sugars are pentoses. When the sugars are fused with the nitrogenous bases, there is the formation of another compounds, or we can say when sugars fuse with the nitrogenous bases, they result in the formation of another compound. For example, when adenine fuses with the pentose sugar, it doesn't remain the adenines still more, it becomes the adenosine. When hydrogen reacts with the oxygen, what it forms? Water, H2O. So it doesn't remain still hydrogen and oxygen, new compound is formed. Similarly here, when pentoses are fused with the nitrogenous bases, they result in the formation of another compound. Here adenine fuses with the pentose sugar, it results in the formation of adenosine. Guanine fuses with the pentose sugar, it results in the formation of guanosine. Cytosine fuses with the pentose sugar, it results in the formation of cytidine, not cytosine, cytidine. And similarly, when thymine fuses with the pentose sugar, it results in the formation of thymidine. So still we are not at the position to make the nucleotide. We have to study first the nucleotide, because when we saw the definition of DNA, it is a linear polymer of nucleotides. Linear we have got, polymer we have got, now remaining is nucleotide. Still we are not in the position to make the nucleotide. So we have fused the nitrogenous bases with the pentoses, right? When we fuse the nitrogenous bases with the pentoses, right, it results in the formation of nucleosides, not nucleotides. Nucleosides, it results in the formation of nucleosides. So we need to convert the nucleoside into nucleotide. What we are going to do, we are going to add the phosphates. phosphate is an energy molecule. Whenever we are going to add the phosphate to any compound, it results in the release of too much energy. For example, we have adenosine. Adenine plus pentose sugar results in the formation of adenosine. So we have adenosine. We are going to add one phosphate to the adenosine. What it will form? Adenosine monophosphate. Going to add the two phosphate atoms or elements. What it will make? It will make the adenosine biphosphate or diphosphate. When we add the three atoms of phosphate to the adenosine, what it will make? Adenosine triphosphate. That you know, that is the most energetic molecule present in our body. So how much phosphate atoms you are going to add, that much energy is going to release. So here we need to add one phosphate so that we can make the nucleotide. So we have nucleoside. To nucleoside we are going to add one phosphate molecule at the fifth carbon atom. When we are adding the phosphate atom at the fifth carbon position, it results in the formation of nucleotides. So how the nucleotide is formed? Nitrogenous base plus pentose sugar plus phosphate molecule gives nucleotide. Now the nucleotides are formed. We need to make the bond between the nucleotides so that we can make the strand of DNA. One strand of DNA we can make by binding the nucleotides in a chain, in an unbranched chain. Okay, so when we are binding the nucleotides, you know, anything which is going to bind, it needs some energy for the bonding. So here what's happening actually, when nucleosides get converted into the nucleotides, that time one phosphate is added. Actually it's not one phosphate, it is actually the triphosphate that's added to the nucleoside and results in the formation of nucleotide. And at the time of bonding between the nucleotides, two phosphates get detached from the nucleotide, and that detachment of two phosphates results in a release of energy, and that energy is used by the enzymes. Which enzymes? Those enzymes who will help the nucleotides to make a bond between them. Okay, so enzymes are using the energy for making the bond between the nucleotides. So when the enzymes are using the energy for making the bond between the nucleotides, so it means at 5 prime, at 5 position, 5 carbon position, that's what we are calling 5 prime later on. At 5 carbon position, two phosphates are getting detached, two phosphates are removed, and those two phosphates results in the release of too much of energy, and that energy is used by the enzymes for making the bond between the nucleotides. Now we need to add the nucleotides. Okay, suppose this is one nucleotide and this is another nucleotide. This nucleotide has to get attached with this nucleotide. When this nucleotide is going to get attached with this nucleotide, it attaches at the carbon number 3rd position. It attaches at the carbon number 3rd position. So second nucleotide attached to the first nucleotide at the carbon number 3rd position. Third nucleotide attaches the second nucleotide at 3rd carbon position. So it continues and makes a one long chain, and that results in the formation of one strand of DNA. The bond that's formed between the nucleotides is known as phosphodiester bond. The bond that is formed between the nucleotides is known as phosphodiester bond. Now this bond is formed between the nucleotides. Now I will tell you one thing, this bond can also be broken. This bond can be broken by the enzyme, and that enzyme is known as nuclease. Nucleases are the enzymes that are used in the breakage of bond between the nucleotides. Okay, so nucleases are breaking the phosphodiester bond that is formed between the nucleotides. Now those nucleases that break the bond between the DNA are known as DNases. DNases. Now those enzymes that breaks the bond between the DNA is known as DNases are known as DNA. There are different nucleases. Some nucleases breaks the bond at the terminal points, at the end points, and those nucleases are known as exonucleases. Those nucleases that breaks the bond at the terminal site are known as exonucleases, and those nucleases that breaks the bond in between the chain, those nucleases are known as endonucleases. There are the bases, nitrogenous bases, and they are complementary to each other. Like adenine is complementary to thymine, not thymine. Adenine is complementary to thymine. Guanine is complementary to cytosine. Adenine makes double bond with the thymine, and guanine makes the triple bond with the cytosine. So these bases that makes the bond between themselves, they are known as complementary bases. So adenine is complementary to thymine, guanine is complementary to cytosine, so they are the complementary bases. So that's all about the DNA structure and DNA formation. Next, we will see based on your suggestion also, and hope you all can understand my lecture. And if you have any doubt about my lecture about this DNA structure and DNA formation, you can ask me directly in the comment section, and definitely I will reply to each and every one. It's my promise. Okay, bye.